DMD, or Duchenne muscular dystrophy, is a progressive disease characterized by muscle degeneration and weakness. DMD is caused by a genetic change or mutation in the gene responsible for making a protein called dystrophin. The mutation in this gene causes a dysfunctional dystrophin or even the omission of dystrophin from muscle. The lack of a working dystrophin destabilizes the muscle fiber membranes, allowing it to become prone to chronic injury, inflammation, and eventual degeneration. Over time, the muscles will become so weak that a wheelchair for the patient will become necessary. By age 20, cardiac or respiratory failure becomes a real danger as the muscles become too weak for simply breathing or pumping blood to the body. There are many avenues of approach for a treatment to DMD, but I want to talk about one interesting approach that is more obscure but has great potential: utrophin upregulation via CRISPR-Cas9 technology. Increasing utrophin levels, a protein very similar to dystrophin, is known to help recapitulate healthy function of the muscle in mouse models. And CRISPR-Cas9 technology has been established as a method for targeted genome editing. It has also been repurposed for gene modulation, i.e. the activation or repression of certain genes. This can be a powerful tool to help all kinds of patients with all kinds of diseases. So the idea is to use a modified CRISPR-Cas9 system in order to enhance DNA expression of utrophin in DMD patients, so that utrophin can replace dystrophin in muscles and hopefully restore normal muscle function. You now see that CRISPR is important. But what is CRISPR exactly? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is important to note that there are several types of CRISPR, but we will be focusing on type 2, also known as CRISPR-Cas9. So how does it work? Let's break it down. The type 2 CRISPR gene codes for these essential things: a Cas9, or CRISPR-associated protein 9 nuclease, which is a type of enzyme responsible for making cuts in DNA, a pre-CRISPR RNA. This RNA will contain regions called spacers derived from foreign DNA, and a trans-activating CRISPR RNA. The pre-CRISPR RNA and the trans-activating CRISPR RNA then hybridize and are processed by Cas9 and RNase 3. The CRISPR RNA and trans-activating RNA duplex is called a single guide RNA. The single guide RNA and Cas9 complex then searches for DNA that matches the spacer on the single guide RNA. If a matching sequence is found, the single guide RNA will bind to the complementary sequence on the target or foreign DNA with the help of the protospacer adjacent motif, or PAM. Cas9 then cleaves DNA 3 bases upstream of the PAM site, creating a double-strand break, or DSB, with blunt ends that cannot recombine without cellular repair machinery. So the result of this process is the targeted acquisition of a specific DNA sequence and its subsequent cleaving. How was CRISPR discovered? Francisco Mojica, a Spanish researcher, was the first to identify the CRISPR gene location back in 1993. After several years of research, in 2005, he hypothesized about its function as an adaptive immune system. The discovery of the components of CRISPR-Cas9 that we talked about took almost a decade, and the contribution of several groups of scientists. Alexandre Bolletin of the French National Institute for Agricultural Research discovered Cas9 and PAM in May of 2005. John van der Oost discovered the CRISPR RNA in 2008 in the Netherlands. In 2011, the group of Emmanuel Charpentier discovered the second RNA trans-activating CRISPR RNA for the CRISPR-Cas9 system. This was the final piece of the puzzle for understanding the mechanism. Later, she and Jennifer Doudna had successfully demonstrated CRISPR-Cas9 in vitro. In 2013, Feng Zheng at MIT was able to harness CRISPR-Cas9 technology for genome editing in human and mouse cells. This opened the door for CRISPR-Cas9 to be used in disease therapy. Scientists LaRussa and Chi have stated that CRISPR technology is ushering us into an unprecedented era of biological control, referring specifically to the ability to use CRISPR for activation and inhibition of genes, as well as gene editing. How can CRISPR-Cas9 be modified for genetic activation? First, Cas9 needs to be deactivated because we don't want it cutting DNA anymore. Deactivated Cas9 is also called dCas9. We can then add effectors to the deactivated Cas9. Effectors are molecules that can cause transcription activation or repression. They are called activators or repressors, respectively. For example, in 2013, Picard et al. Was able to fuse the omega subunit of RNA polymerase to deactivated Cas9. A deactivated Cas9 plus an activator complex can be used with single guide RNA to act as a programmable artificial transcription factor. The single guide RNA is our GPS. We still need it to locate our gene of interest, in this case, the utrophin gene. This is known as CRISPR activation. You may be asking why not just use the gene editing capacity of CRISPR-Cas9 to correct the actual dystrophin gene. And the answer to this is that it allows us to avoid double-strand breaks, or DSBs, that could cause unwanted mutations and permanent genetic damage. Deactivated Cas9 and an activator domain called VP160 was used in one experiment by Wojtal et al. In 2016 to target the utrophin gene in vitro. The cells used were immortalized myoblasts from a DMD patient. Electroporation was used to directly introduce the deactivated Cas9 to VP160 genes with the appropriate single guide RNA. Results are promising, indicating up to a 6.9-fold increase in utrophin from basal levels. Beta-dystroglycan levels also increased, providing evidence for the functional relevance of the strategy. Another study by Xin-Kai Liao et al. In 2017 had achieved utrophin gene activation in vivo using epigenetic modification. How do they accomplish this in vivo? They used adeno-associated viruses, or AAVs, to deliver the CRISPR-derived genes to the MDX mice model. AAVs are the most promising method of gene delivery in vivo. These AAVs are small, however, so some changes need to be made for everything to fit. First, the activator molecules were separated from Cas9. This was accomplished via the synergistic activation mediator, or SAM, which recruits an activator complex called MS2P65 HSF1, or MPH for short, at the gene site. Second, smaller single guide RNAs were made called dead single guide RNAs. These dead single guide RNAs prevent Cas9 from performing its nuclease function, so the use of deactivated Cas9 becomes unnecessary, and wild-type Cas9 can be used. Note that the dead single guide RNAs were modified to allow the MPH complex to bind to it. Xin-Kai Liao et al. Also used Cas9 expressing mice to overcome delivery challenges of the enzyme. Using the AAV dead single guide RNA MPH method, they were able to induce utrophin expression in mice compared to control, and exhibit improved hind limb muscle strength 2 months later. They also showed that the histological disease manifestations in MDX mice were reduced even after the pathophysiology had been established. Specifically, they were able to show that muscle fiber sizes increased along with the frequency of total muscle fibers. This is the first system to induce physiologically relevant phenotypes via CRISPR-Cas9 mediated epigenetic editing, and this was back in 2017. CRISPR-Cas9 is an awesome toolkit that researchers are beginning to take full advantage of for the treatment of diseases like DMD. More preclinical studies using genetic and epigenetic modulation via modified CRISPR-Cas9 technology are needed. In the future, I hope to see our generation expanding upon CRISPR-Cas9 modulation and even discover new and exciting technologies for gene modulation and editing. I hope you enjoyed this video, and thank you for listening.